Okay, good morning and welcome. So let's start. Let's open this conference. So welcome everybody. I'm Elena Demomi and I'm honored to be here to open this IRIM conference, IRIM 3D. I'm here with my co-chair, Professor Arricchiello, and I'm really happy and honored to be here to open the conference. So first of all, a word of thanks to the other co-chairs, so the organizing committee, who helped a lot during the last past months. So in particular, I would like to thank Program Chair Calogero Oddo, who is here, the workshop chair, Professor Costante, who is there, and the other guys who are around the rooms and around the conference premises still to fix issues and so on. So the local chair, Andrea Mofonti, financial chair, Federica Ferraguti, here with us, the sponsor co-chair, Professor Jonathan Salvietti, the challenge chair, which you can meet in the challenge space in the exhibition in the Maker Fair, the award chair, Clemente Lauretti, then the award chair, Professor Riccardo Costanzi, communication chair, who have worked a lot on the LinkedIn pages and the advertisement, Maria Pozzi, the Dreams activity chair, who coordinated the activities of the PhD Dreams, Professor Matteucci, and the exhibition chair, who you see all around the conference, fixing issues and open points, who is Francesco Scotto Di Luzio. All the chairs were decided by great people who helped a lot for the program, the workshop, and so on, who you can see here in the list. Also, I would like to spend some thanks word for the sponsors who provided a lot of help for the conference organizations. First of all, Maker Fair, which is hosting us here. The patronage from the RAS chapter of Italy, and the sponsor which provided awards for the publication and for the papers that will be given tomorrow evening. So Fit for Med, PAL for the challenges, Oversonic, Endoteranostic, Pariter Partners for the coffee break sponsor, and Hotel Villa Vanda, who kindly sponsored the Dreams activities. Now I leave the stage to my co-chair.
Good morning, everybody. Okay. Good morning, everybody. I'm Filippo Arricchiello. It's also an honor for me to host you and to welcome here in this location. I would start with just an explanation of the program that is quite articulated and complex. So we have 2 days of conferences in the area of the ISA, while we have a 3 days event in the Maker Fair area. In this area, so in the 2 days you will see in this area both the keynotes, the opening, the plenary events, the robo pitch, and the events of the PhD Dream that will be given in the area of the ISA. While in the 3 days in the area of the Maker Fairs, you will find the exhibition. There will be a set of stands of the Maker Fair related to the keywords of robotics that are related to IRIM, and also the challenges will be organized in that area. You will have a separate ticket. You already have a separate ticket to access to the area of the Maker Fair. You receive the ticket by email, and you have to use to enter from the other entrance. Also, for the logistic, now we are in the building of where there is the Aula Magna, in which we will have all the plenary events, while other part of the conference will be held in the other building, that is the building F, in which we will have 2 different floors. At the ground floor, you will find the Sala Caravaggio, in which we will have all the lunch, coffee break, and workshop. And also the poster session will be at the ground floor of the building F. While at the first floor, you will find the 3 rooms that are also used for the workshop. In the program on the website, you will also have a link to the Easy Share, from which you can have the details of all the events, all the location of the separate events. While to enter the area of the competition and the exhibition, you have to go out from the entrance and enter from the general Maker Fair access to the area. Today, we will start with the keynotes. Also, we have keynotes and roundtables both today and tomorrow. Today, we will have the keynote from Pietro Valdastri and from Emanuela Girardi, and also we will have one roundtable on founding dilemma in robotics, and this will be held in the Aula Magna here. While tomorrow, we will have other two keynotes lecture from the Alessandro Asciutti and Antonio Bicchi, and also we will have another roundtable about humanoid in robotics. Now, let's start with the workshop. I will leave the floor to Gabriele Costante, that is the chair of the workshop.
Thank you. Thank you very much. I would say it's an honor also for me to be the workshop chair. I would like to thank Elena and Filippo for this great opportunity, actually. And I also would like to thank all the others co-chairs that helped me a lot. They did an amazing job in assisting me in the proposal for the workshop and managing also the review process. So Dario Calogero Guastella, Martina Lippi, Massimiliano Mancini, Francesco Visentin, and Mariangela Filosa. I don't know if they are here, but thank you again for all your great help. And I would also like to thank all the community for the many contributions we received for the workshop proposals. They were all outstanding proposals. And so we will have a lot of these papers and proposals for during the workshop session in the next days. Workshop sessions are divided into 3 main sessions with 5 parallel workshops and 4 parallel workshops on the Saturday session. So this morning we will start with 5 parallel workshops. You can see on the green tags here. Then we will have in the afternoon session additional 5 workshops in the orange tags. And finally 4 sessions tomorrow during the afternoon in the blue tags here. The workshops are in the 5 rooms, Aula Magna, Sala Caravaggio, and room 19, 20, and 21. And you can recognize the assignment to the workshop by looking at the tags and the papers that are attached on the doors of each room, and also by looking at the online program. And for details on about how they are organized, I will leave the stage to Professor Oddo, which will give some details about that. Thank you.
So let me remark that one analysis that was made in the previous editions of IRIM was somehow the not integrated program between the workshop and the contributions that are submitted resulting from call for paper. Instead, this year, as a committee, we agreed to make a modification. Of course, we'll discuss maybe in the assembly whether this modification is a good improvement or maybe we have to go back to the previous organization. This year, we decided to attach the contributions to the poster. This way, to the workshop, sorry. So this way, the workshops are fully part of the program, and the contributions resulting from submissions are linked to the workshop initiatives that are, let's say, less referring to the objectives only and the topics only of the individual organizers or the group of organizers of each workshop, but are part of larger initiatives. So each submission resulting from the peer review process and evaluation with the committee was attached to the workshops that were reviewed previously. And here you see the program. So in those submissions that applied with the call for paper, you will present your pitches at the end of the related workshop. So you find the correspondence in the program. So you will have 2-minute pitches at the end of the most pertinent workshops that we identified or that you identified, the submissions identified at the time of the contribution proposal. And then you will participate in the poster session immediately after the related workshop. So operationally, let me give and I conclude some indication for those contributors that have a workshop, that have posters at the end of each workshop. Please, before the start of the workshop, go in the poster area and attach the poster. You will find poster holders with numbers that are related to the specific workshop to which your contribution has been attached.
Okay, let's go on with the other events, so the other part of the programs. This afternoon, there will be also the RoboPitch, that is an event dedicated to the startup in robotics. There are 5 startups that have been chosen to give a pitch, a presentation of their project at this early stage, their proof of concept, to an audience done by venture capitalists, by research personnel from the university, from the industry, that can evaluate their initial business proposal, and then there will be also an award for the pitch. And this event is organized by Alessandro Settimi, and here you can see the list of participants this afternoon in the Aula Magna about the RoboPitch. Then there will be other 2 events related to the PhD Dream school. In particular, tomorrow there will be a meeting that is PhD Turn 40s, in which the young PhD students will have the possibility to discuss, interact with the professor that took the PhD several years ago, and is a way to make 2 different generations of roboticists to interact one with each other. So it will be a good opportunity for the PhD students to interact with the major roboticists that we have. And also there is another event related to the PhD school, in particular the students of the PhD Dream program that are at the 3rd year, they will do the final exam to be admitted to the final defense on Sunday morning, and there will be the committee will judge the students here. So also on Sunday morning, only for the PhD Dream will be this part of the event in the ISA area. Then there will be, in the area of the Maker Faire, there will be also 2 challenges. In particular, there is a challenge organized on the topic of navigation, while the other one is on human-robot collaborative manipulation. The 6 teams of researchers will compete for the challenge. The challenge will be in the Maker Faire area. There will be, on the 3 days, the last day on Sunday morning, there will be the final race, and there will be the setting of the award around the lunchtime. So if you are still here on Sunday morning, be there around 12:30, and you will see the final part of these challenges. The challenges are in the area of the Maker Faire because it's a part of the idea of how IRIM should interact with the more open community. In the Maker Faire, you will find students, kids, families, hobbyists. So it's a way in which our community can interact and show part of the activities to the more general audience. Then there will be also in the exhibition, still in the area of the Maker Faire, there is a large number of stands from university, from research center, from Power Robotics, that is one of the sponsors, and also the competition will be done using a robot from Power Robotics, stand for Fit for Med. And all this stand related to IRIM you will find in that area. If you have the time to go on the Maker Faire, you will see a set of stands with the logo of IRIM that are all the stands related to this conference. About the social event, this late afternoon we will have the welcome cocktail in the Sala Caravaggio, so in the ground floor where we also have the lunch. While tomorrow evening we will have the social dinner that will be at the Bahia restaurant, that is a nice boat on the Tevere River. And the events will be there. There will be, during the dinner, there will be also given the award. We have a large list of awards that are provided by the large number of sponsors that was found by our sponsor committee. And you can see that there are awards for the poster, for the workshop, for some paper on specific topic, about the challenges. So this year we will have a large number of awards, and there will be a final presentation at the end tomorrow around 6:30, while the award will be given during the social dinner. About the communication, you have seen a lot of posts on LinkedIn, also on on the mailing list of the community. Here there is a QR code that I invite you to frame the QR code. This is for the WhatsApp community of the conference. So you can receive during these 3 days of activities some information about logistics on what is going on. So you can frame the QR code here. It's also in the final slide. I will leave for a few minutes this QR code.
It's in the bags.
It's also in the bag, so you already have the QR code. Okay, about numbers, just the last few slides. About the events of the IRIM, we will have 4 keynotes, 2 roundtables. We received 17 submissions for workshop, and just for logistic issues, we were able to accept only 14 of them. We received 188 abstract submission, and we accepted 103 abstract, and that will be presented in the form of poster. We have a RoboPitch event with 5 startups, 2 challenges with 6 teams, 2 PhD team events, the exhibition, and also you will find around a large number of volunteers that are the guys with the yellow t-shirt that can support and help you in the logistics to do not get lost in this area. Finally, the numbers about the event. We have 127 full registration. Large part is from PhD students, and we have 50 one-day registration. Most of the registration for the one day are for today. Less for tomorrow. We know that there is an overlap of dates with the AROS conference, so I know that a lot of person will leave tomorrow. We have 6 person with the registration of the challenge, and finally we are 183 registration. The financial chair is quite happy, Federica Ferraguti. This is the last slide. You can find the WhatsApp community code and also the conference website, the QR code for the conference website. From the conference website, in the page of the program, there is a link to EasyShare, and from there you can get all the abstract, the location of the different workshop, the end of the different presentations. So it's a useful link. Okay, I guess we can start. I will leave the floor to Federica Ferraguti that will introduce the first keynote lecture from Pietro Valdastri. Thanks.
Ci stiamo? Okay, thank you very much, and good morning, everyone. Thank you for your great work in organizing this conference. It's my great pleasure to introduce the first speaker of today and of the conference, which is Professor Pietro Valdastri. Pietro is a full professor and chair in robotics and autonomous systems at the University of Leeds. He directs the Science and Technologies of Robotics in Medicine lab, focusing on intelligent robots to fight cancer, the Institute of Robotics, Autonomous Systems and Sensing, and the Robotics at Leeds network. Professor Valdastri served as principal investigator on grants exceeding $27 million. His significant awards include the NSF Career Award in 2015, the ERC Consolidator Grant Award in 2019, and the KUKA Innovation Award for his robotic colonoscopy platform in the same year. He co-founded WIM Medical, which was acquired in 2017, and recently started Atlas Endoscopy Limited to commercialize his robotic colonoscopy platform. So please, Pietro, the floor is yours.
Thank you, thank you. Thank you, Federica, and thanks to the organizers for inviting me to this event. So it's I mean, of course, I'm Italian. I grew up academically at Scuola Superiore Sant'Anna, and it's great to see a lot of young faces coming to this event. And I have to say, I mean, I've been around the world. I've been 5 years in the US, and now more than 8 years in the UK. I have to say that Italian roboticists are among the best. So our nation is very, very well represented in robotics, and so keep it up with the great work that we are doing in Italy. Okay, so first of all, a disclosure. As Federica mentioned, I co-founded Atlas Endoscopy, and I'm also the CTO. So I will make some statement about robotic colonoscopy that you have to take with a pinch of salt, because I'm definitely biased. Okay, so my research is mainly on creating new robotic platforms from scratch, and what we like to do in my lab is to use magnetic actuation, so magnetic fields to control medical robots. And today I will tell you about the platform we developed for robotic colonoscopy. And I want to focus your attention on the fact that bringing a platform from the lab to final fruition, especially in medical robotics, it takes time. And so what I'll tell you about is a story that started back in 2008. This platform is probably the one I've been working on the most. More recently, we started working on magnetic catheters, and even more recently, we started on magnetic vine robots. So today, since my time is limited, I will talk about the platform for robotic colonoscopy, and then I'll give you an idea of what we can do with magnetic vine robots, but I will skip the part in the middle. But if you are interested, then come and see me after my talk. Okay, so before we start, I want to just point you to a review paper that I was fortunate to co-author a few years ago about medical robotics in the last decade. So in this review paper, me and co-authors analyzed several fields like robotic surgery, robotic endoscopy, exoskeletons, medical capsule robots, and so on. And we basically listed some of the most relevant papers in the last decade. But in doing this, we also analyzed different levels of autonomy in those different fields. And so as roboticists, I'm sure you are aware about the challenge that we are facing in trying to make robots more and more autonomous, adopting the same paradigm that we have now in automotive. So no autonomy, old style, old-fashioned car, full autonomy, self-driving car. And that is something we are embracing also in the community of surgical robotics and medical robotics, where we want to move from pure teleoperation, like the surgical robots that are currently in hospital, to more and more increasing levels of autonomy, so to reduce the mental burden on doctors and also increase capacity. And so today I will try to show you some examples of how we can move in terms of increasing autonomy in the field of robotic colonoscopy. And so, talking about robotic colonoscopy, why is that important? So, the main disease we are after is colorectal cancer. So colorectal cancer is the second leading cause of cancer-related death. So it's a big killer. Is the cancer in the last part of the gastrointestinal tract, which is the colon. It grows in 5 to 10 years, starting from a small lesion called polyp, and becomes more and more bigger and bigger, up to stage 4, when it becomes metastatic and spread in the body of the patient. So what is interesting is that if this type of cancer is detected at an early stage, it can be removed. Just removing the tissue there in the colon removes the cancer in its totality. And so there is mostly 100% of 5-year survival rate. So if the cancer is detected at that stage, the patient has no problem in the future. At the contrary, if the cancer is detected at stage 4, the 5-year survival rate drops down to 10%, 11%, and in that case, the patient needs chemotherapy and mostly invasive procedures. So it's extremely important to screen for colorectal cancer. So important that, for example, in the United States, screening starts at 45. So if you are older than 45, you should think, maybe I should have had my first colonoscopy. What is a colonoscopy? So colonoscopy is what, for example, again, in the US, they use for screening. Here in Europe, is more like fecal blood test, which is the amount of blood found in the feces, and if that test comes out positive, then you get a colonoscopy. In the United States, you go straight to colonoscopy because it's a more effective, although more expensive, but also more effective procedure for screening, because if you see the lesion, you can remove it straight away. And so colonoscopy consists in the introduction of like 1.5 meter long, quite stiff tube inside the colon of the patient. And this tube has to travel, let's see if yeah, can you see my, yes. This tube needs to travel from the anus, up, and then down to the cecum. This tube has a camera at the tip, the possibility to articulate the orientation of the camera with cable steering, and those cables are connected to the knobs here at the user interface. There is illumination, and there is insufflation to create space, and there is also an instrument channel so that an instrument can be used to either get a biopsy or remove tissue. Typically, and this is just to mention how it works, typically the doctor introduces the endoscope and gets to the cecum, to the end of the colon as fast as possible. Average time is 5 minutes. And then, while going back, look around to see if there are lesions. Now, colonoscopy is a very effective procedure, so it works well, but unfortunately it has a lot of drawbacks. And all those drawbacks are related to the design of the endoscope. That, as you can see here, the endoscope we use, the colonoscope we use these days is quite similar to what was the first endoscope, flexible endoscope, in 1957. So imaging has changed a lot. In 1957, it was a bundle of optical fibers, and so the doctor had to look into the ocular to see the image. These days is a chip-on-tip camera, super high resolution, so the image really improved. But the idea of introducing a stiff tube inside a convoluted and soft anatomy is the same. And so what happens if you introduce and push a stiff tube inside a soft anatomy? You go stretch the tissue, as you can see in this animation. So there is no way you don't stretch the tissue, and this stretching of tissue causes pain. And so colonoscopy is a very painful procedure for the patient. So much so that most of patients, except for example in the United States, they get full anesthesia, full sedation. And this is, of course, not ideal. We would like to have a painless procedure. Also, colonoscopy is extremely unintuitive for the operator. And so the training to become a proficient gastroenterologist and perform colonoscopy is very long. And this is because the user interface is not ergonomic at all. The mapping of the movement and the visual feedback is really tricky. And so something like an improvement here is also really needed. And also the instrument is expensive. So it's about 50,000–60,000 euros. And so it must be reused, of course, to make sense. And so colonoscopy is only limited to large hospital or large endoscopic center where the endoscope can be reprocessed and cleaned in between cases. And so this limits capacity for colonoscopy, and especially in places like the United States where after 45 everyone is getting a colonoscopy. That means there are 22 million colonoscopies every year, but there is no capacity. So there are not enough trained operators, and there are not enough centers that can provide all those colonoscopies. And so there is real need for some new technology. And through the years, the robotics community has been very active in trying to find a solution to this problem. So this is a review paper mapping some of the state-of-the-art in robotic colonoscopy. Interestingly enough, the only one robot that is FDA approved, CE marked, and available in hospital is this one here. This is the Endotics system. It comes from a company that started in Pisa, out of Scuola Superiore Sant'Anna, and it involves a robot that works with inchworm locomotion. So basically, this robot is a flexible robot, has 2 clampers and a body that can elongate and retract. And so it can clamp at the back, elongate the body, clamp at the top, release the back, and retract the elongator, and so on. So it basically finds its way as a worm inside the colon of the patient. So with the idea of this being completely painless. And so I passed 45 years of age, and since I'm designing robotic colonoscopy platforms, this year I said, okay, I probably need my first colonoscopy, and I wanted to do some industrial espionage, and so I forced myself to have a robotic colonoscopy. And so here I'm holding this colonoscopy robot at the hospital in Pisa, and this is me on the inside. And so I wanted to see the user interface. I wanted to see if really was painless, if it needed to be improved. And actually, yes, it definitely needed to be improved, because the time for this robot to reach my end of the colon was 57 minutes. It was a painless procedure. So it was supposed to be painless, so I was not sedated, I was not anesthetized, so I was fully awake and take it moving. And at the beginning, I was excited to see a robot in action, and then it was a disaster. It even broke inside myself, the robot. So it broke in a non-catastrophic way, because this is a CE-marked product, so it is safe. But what broke was one of the clamper, basically has a defect at some point, and so was not able to operate anymore. And so it was just not working. And so my colonoscopy went well, in a sense. I didn't have any problem, but this robot really is terrible. And again, this is my view, just one data point, so take it with a pinch of salt. Anyway, there is still room for improving in the field. And so that makes me glad, because we are developing an alternative robotic platform, which is based on magnetic fields. So the principle we are applying is something that we explored when I still was assistant professor at Scuola Superiore Sant'Anna. We were involved in a European project, Framework Program 6 European project, so back in the days, called Vector. And yeah, we developed a platform that has a robotic arm, a big permanent magnet, and a small permanent magnet at the tip of the flexible endoscope. And then all the other parts are the same as a traditional colonoscope, so camera, illumination, instrument channel, and so on. The main difference is that we don't need to have a stiff body anymore, because we have magnetic pulling of the tip. So we are applying the force to move the endoscope right at the tip and not at the back, so we need way less force. And also we orient the tip of the camera, the tip of the endoscope, by magnetic fields. And so this allows us to be extremely flexible in the body of the system, and that we hypothesize this platform to be painless. We also have robotics, so we believe we can make it as intuitive as possible, like driving a car in a video game with a joystick. And also this is an ultra low-cost device. So the bill of material of the flexible endoscope itself is about 10 euros. So we can make the device disposable, and so we don't need any more cleaning and reprocessing of the scope. So ideally, we can put this robot in a van and go in rural areas and do colonoscopy in a van, kind of scenario. Okay, but this is all based on magnetic manipulation. And so one slide about how this works. So magnetic manipulation, if you have a big permanent magnet outside the body and a small permanent magnet inside the body, you can move the small magnet leveraging the gradient, while orient the small magnet leveraging the misalignment in field. As you can see from this picture, the magnetic field is highly nonlinear in space, and so it's not an easy task for a roboticist. And so something which is crucial and then really enable us to make this work in an intuitive way was real-time localization. So during my 5 years at Vanderbilt University in the US, my team developed what is now the core technology of our platform, real-time post-detection. So we have a number of magnetic field sensor and inertial sensor at the tip of the endoscope, and those allow us to track in 3D, in 6 degrees of freedom, position and orientation, the tip of the endoscope in a workspace which would host would allow a patient, and with a refresh rate of about 1 kilohertz. And so with this information, we now can decide how to move the external magnet, to position the external magnet, so we get the user intent, or we accomplish the task that we want. So again, localization, crucial for working and closed loop control in real time and make all of this intuitive. And so with this, we started to explore different level of robotic automation, starting from robot assistants. So basically, in this case, the user is looking at the image coming from the camera as a joystick, and the joystick, moving the joystick up, down, left, and right with respect to the camera frame, tells the robot how to move in combination with localization, of course. And so we did a set of experiments with 10 different users, 5 repetition each user, and we were able to navigate a colon phantom in less than 4 minutes, which is the average, similar to the average time that is required to complete a colonoscopy. So we were very happy there, and now we moved a step forward. So this is what we are implementing in the commercial platform from Atlas Endoscopy. So we have a lumen tracking algorithm that tells us where the lumen is, and if the user is losing the lumen from the image, an arrow shows where the lumen was, and so that the user can reposition in that direction. We can also do task automation. So for example, we can automate the task of retroflexion. Retroflexion means from looking forward to looking backward. And this is important because sometimes polyps are hidden behind tissue folds, as you can see here in the top left. And so having the possibility to look behind is very important. And this is not possible with the conventional colonoscope because it's too rigid. And so for us, we push a button, the system localizes the tip of the endoscope and decide how to move the external magnet to get retroflexion. And we can get retroflexion in about 10 seconds. So this is an example of task automation. Another example is support for when we use an instrument to get a biopsy sample. And so once we reach the end of the colon, we just pull the we just pull the flexible endoscope, the external magnet localize it and follow to maintain magnetic coupling. If a target is identified, whether with an AI algorithm or by the doctor, we can say, I want to take a biopsy sample of the target. And so the system look at the target from 2 different angles and evaluate what is the distance, and then aligns the point where the instrument would come out from the instrument channel to the target and keep stable this position in closed loop control. And so the operator can drop the joystick and start using the instrument. And this, just to mention, this is a procedure that typically involves 2 people in the endoscopy room, one aiming the colonoscope and the other one operating the instrument. With our robotic platform, it just takes 1 person. And so this reduces the number of people required in the endoscopy room. Another thing we are working on is 3D colon reconstruction, and this is also a collaboration with Professor De Momi at Polytechnic of Milan. So since we have localization data related to the image coming from the camera, we have a good chance to reconstruct the colon in 3D, and this is very important because we can tell the operator that if we have visualized all the colon, and we can also say, okay, there are blind spots, go back and check those back again. And we can also certify that the colonoscopy has visualized all the colon, which is something that for screening is very important and is not available at the moment. Another task we try to automate with a variation of our flexible endoscope is ultrasound scanning. And so we wanted to place an ultrasound array at the side of the flexible endoscope to scan the mucosa of the colon. And this would basically replace the need to get a biopsy, because if you see something suspicious, you typically get a biopsy, send it to a pathology lab, and after 2-3 weeks you get a response, and then if it's positive, so you need to call back the patient, find back the same spot, and operate. While if you can decide on the spot with ultrasound, for example, if the polyp, if the lesion is cancerous or not, you can remove it straight away. And so we wanted to integrate this ultrasound capability, but to do that, we need to control the roll, the degree of freedom of roll motion. And this is something that is impossible with magnetic manipulation, because the magnet inside the tip of the endoscope is a cylinder, axially magnetized, and is axial symmetric around his main axis. And so you don't have the possibility to roll by modifying the external magnetic field. There is no way to do that. And so one of my PhD students, she's very bright, now she's graduated, she came with the idea of changing the shape of the magnetic endoscope. And so she found this specific shape, which is called oloid, and it's also like a developable roller. And so it's a shape that if you just translate it on a plane, it would expose every part of the surface to the surface that is touching. And so we took the idea of the oloid, modified the shape of our flexible endoscope, and you see in this case, maybe here, yes. So we had an ultrasound array, and on this side, the shape of the oloid. And so basically in this case, by adapting the motion of the external magnet, we were able to recreate a roll motion thanks to the shape of the flexible endoscope. And so here we had two copper lines, here and here, and we first create contact, and then we are able to do this sweep motion and visualize the copper wires in the ultrasound image. And we also have an indicator of the quality of the contact pressure, which is given by the ultrasound signal. And so here you can see, this is an animal experiment in pig, and we demonstrated we are able to do rolling and sweeping, thanks again to the interaction of this new shape. Well, one would say it's embodied intelligence, but it's a fancy shape combined with magnetic manipulation. And then we tried to see if this worked for a relevant example. So we injected a specific quantity of saline solution underneath the mucosa to recreate a polyp or a lesion. And then we did a scan of this region, and we were able to reconstruct the pocket of saline solution underneath the mucosa layer. And also by elaborating this 3D image that we were able to acquire, we were able to confirm the amount of saline solution that we injected. And so this is sort of spin out of our magnetic endoscope with an ultrasound transducer at the side. Going back to the mainstream research, then we can also do high level of autonomy. So in this case is full autonomous driving. So the system is again, lumen detection algorithm, tell the system where the center of the lumen is, and the platform is driving the endoscope in that direction. And again, here the user is just left with feeding the tether, so there is no control, it's just extremely simple for the user. And again, in this case, 10 users, 5 repetitions each, we were able to get to the end of the colon in 4 minutes. And so the vision of this would be one single gastroenterologist overseeing 8 to 10 endoscopy rooms, each room with a patient, of course, and a nurse, and the procedure going on autonomously. And if a lesion is detected, then the gastroenterologist comes in to perform the procedure. And so again, this would definitely dramatically increase capacity for colonoscopy. Here it's still autonomous driving in a pig trial, and you can appreciate how convolute was the trajectory that we had to follow. But then, of course, the next step is human trials. And so about 1 year ago, we did the first in-human trial at Vanderbilt University in the United States. We enrolled 5 patients undergoing screening colonoscopy. So they were doing their exam under full anesthesia. Then they woke up and got our robotic colonoscopy right afterwards. So they were awake, interacting with us, and also being able to tell if the procedure was painful or not. And so the outcomes were that all the 5 patients did not any adverse event, no trauma, and they all tolerated extremely well the procedure. So this is very encouraging for us because it means that maybe we are on to something and that the procedure is really painless. But of course, these are just 5 patients. We need a larger study to confirm, but the preliminary results are extremely encouraging. Where are we now? So as I mentioned, we started a company about 2 years ago, which is called Atlas Endoscopy. We raised some non-dilutive capital with grants and also got 2 rounds of seed investment, and we are now looking for a Series A investment. Unfortunately, we have some interest from the market leader in the field of colonoscopy, which is, again, very encouraging. So you can see in these 2 years, we made big progress from the university platform. In particular, we integrated our own camera. So while in the university platform, we were using an off-the-shelf camera from an Olympus flexible endoscope. And also we made a nice-looking platform with lights and bells and whistle, all the bells and whistle. And it is extremely easy to operate, and it's also fun to drive. It's really like driving a car in a video game. Now I wanted to touch very briefly on how we use also magnetic vine robots. So you are probably aware about vine robots introduced by Stanford University a while ago. And so those robots, they are basically grow from the center by a version. So there is all the material of the robot, which is a soft robot, is inside the robot itself, and applying pressure, this robot grows. And the advantage is that there is no interaction with the environment. There is no friction, there is no sliding. And so we thought about empowering our colonoscopy robot with an hybrid with a vine robot. And so basically for forward locomotion, for forward propulsion using the approach of a vine, and for steering using magnetic field. And this would allow us, for example, to go from the colon into the small intestine, because the more you go deep inside the body, the more friction you have from the tether. And so this idea would allow us to go deeper inside the body. And so we made an hybrid with the system going forward thanks to the vine robot, and steering with magnetic fields at the tip. Again, localization, real-time localization, so we were able to do closed-loop control and so on. But you can appreciate from this video that when we are at bends, the body doesn't interact with the environment because all the material is coming from the center of the robot. And so there is no sliding, there is no friction. And just this was just an example. We are also doing other style of magnetic vine robots, but this is for another talk. Now, I want to conclude by saying, again, it takes time. So this is a picture of me in 2009 with Gastone Asciuti from Scuola Superiore Sant'Anna. We were doing the first trials to see if magnetic colonoscopy was working. And this was, again, 2009, 2011. And now, finally, we are trying to go through FDA approval with this platform. And yeah, it definitely takes time. So be patient, be resilient, and keep it on. So acknowledging all my team, all the funding agencies, especially for this work, NIH, NSF, and ERC. And I also want to pitch that I'm organizing CRAS, which is a conference related to surgical robotics in Leeds in September next year. So if you are interested, please scan the QR code and get in touch. And with this, I'd like to thank you for your attention. I'm happy to answer questions. Thank you.
So thank you very much, Pietro, for your talk. And do you have any questions from the audience? Okay, over there.
About the symmetry for the robot.
Yeah.
Consider for sure that you're not using an asymmetric permanent magnet.
The problem is, the problem is that at a distance, basically you have a dipole-dipole approximation. So if the distance is larger than the magnet itself, even if you have an assembly of asymmetric magnet, it looks like a single dipole. So you still lose the role.
Okay, thank you. And other questions over there?
Okay, yes. So there was, at some point, there was an endoscope that was FDA approved, CE marked, and so on, with one camera in the front and two cameras at the sides. And this didn't work out because you were feeding back to the doctor too many information. And so the mental burden was too much. Maybe in this day, and that time there was no AI, there was no AI polyp detection. Maybe today that can work again, but it was unfortunately a failure. Yeah, there was another platform where they have a camera looking back, coming inside the instrument channel, but this was a failure because you were occupying the instrument channel, which was not ideal. So, yeah. Yeah, Kaloja. So the system is designed to be inherently safe. So if you take in your hand the flexible endoscope and you touch the external magnet, you see that the force is not enough to perforate the tissue, is not enough to get any lesion to the tissue. Because you really need not that much force if the lumen is inflated and you are applying the force where you need it. You don't really need a strong force. So the forces are typically 10 to 100 times lower, like the force you do in flexible endoscopy. And this is something we would like to leverage to eventually get FDA approved for approval for autonomous colonoscopy. It's too big. So to create the same magnetic traction with a coil, you need a large coil and a lot of current. So it's
Okay, I have a question too. And I think that you are a great example of academy that moves to the real world and so to company. What are the problems and how can you address the problems when you talk about automation and certification issues related to this?
So yes, I mean, I think every company, every example has a different story. So for us, we were lucky enough to get the right people. So for me, the team is super important. So if you want to create a startup, you need the right team, you need a team you trust, and you need a team with the right competences. So for us, we were lucky to find the person that got FDA approval and CE mark for the inchworm robot I was mentioning. So this guy left his company and came to work for us, bringing 20 years of experience in robotic colonoscopy and interaction with regulatory, and this is great. So we have been lucky, yes. So, but the team, I would stress the team. Also, like the CEO, you want the CEO you trust because if you start a company, there's so much paperwork, so much legal stuff, and me personally as an engineer, I don't like that stuff. So you really need someone that can cover that and that you trust, because if you don't trust, it's a problem. Yeah.
So we can thank the speaker again. So now we have the coffee break and the workshop will start at 11. So enjoy the coffee break and enjoy the conference.